Okay, yeah, welcome back to this second day of our community workshop. Yesterday we had a great start, and I hope today there will be other amazing discussion. So this is a bit of a reminder of yesterday presentation for the people that joined today for the first time. So what is this workshop about? It's so we want the working group that are currently helping us with the project to connect to each other and with the community. In particular, they are presenting their work and they are posing important questions regarding data sharing in agriculture, and they are gathering feedback from the community about that. So please feel free to join your experience and any of your doubts, even if you are a data curators, data generator, librarian, all of you are very important for us in this project. But before starting, I want to remind everybody that AgBioData is a code of conduct. And so we promote a safe and productive environment that fosters open dialogue and exchange of scientific idea for all participants. This environment is free of harassment and discrimination. If you want to read more about the code of conduct, please visit our website. If you are experiencing or you notice harassment or misconduct and you want to report it, you can send an email to agbioData@gmail.com or to Susan McCarthy at smccarthy.agbiodata@gmail.com. Again, you can find this contact on the website. And with this, I want to encourage everybody to establish an inclusive and respectful environment. So please respect everybody's opinion regardless of their position, encourage them to participate, listen and don't interrupt, and show empathy. And with this is the general agenda. Yesterday, we already started with 3 working groups. For the people that missed yesterday's session, don't worry, after the workshop, I will post the recording on our YouTube channel. So each day we have 3 working groups presenting their work. There are first 3 talks of 10 minutes each, followed by a QA session. Then there is a short break of 10 minutes and then a breakout room session of 45 minutes. Each breakout room has a host, which is a person of the working group that will lead the discussion and will also report at the end of the discussion of the session. There is a scribe, which is a person that takes notes of the discussion. Please, you are free to use the Google Doc that I shared with you in advance, or if you are using other presentation or other notes, please share them with me at the end of the session. And then there is a guardian, which is a member of the steering committee that supervises the breakout room, that everything goes fine, is also a co-host of today. So they will help me with the logistic and can participate in the discussion. And then at the end, the working group report about the discussion during the breakout room with short talk of 5 minutes each. And so with this, I want to introduce today's working group. So we will have the education working group with Meg Staton presenting, the genome assembly and annotation nomenclature with Sarah Dyer, and finally the pan-genome with Gerard Lasso. So I want to remind everybody that if you have any question, feel free to share them in the chat, and I will be happy to ask the question to the speaker at the end of the talk. And with this, I want to invite Meg to share the screen and to start her presentation.
Thanks, Anna Rita. Okay, let's see if
lose Meg.
Meg, are you? We are losing you. Okay. Okay, if Maggie is having issue presenting, Sarah, do you want to start instead of her? And so then Mag will go after.
Meg's back now. Do you want to try again?
Yeah. Meg, you want to try? Do you want me to share the slides and you present them?
Sure. Yeah, let's try that. It did definitely fail as soon as I shared the slides. So maybe if you share the slides, it won't fail.
Okay. Yeah. So let me open them and then I will share them with you. Okay, share. Do you see them? They are still downloading, but you should be able
Yes. Thank you.
Okay, do you see them? Wait, it's loading. Okay.
Yeah, I had the same problem. Okay,
Great.
we can go to the first slide. Okay, so I just wanted to highlight that the education working group is a little bit different from the other working groups, and that we are a separate objective in the NSF grant. And our job is to develop educational materials to train researchers about FAIR data. So we actually will be working kind of with the other working groups that are doing the hard work of building those recommendation standards and implementation plans and making sure that they are part of our educational curriculum. Okay, next slide. We have a great group of people. I'm so excited about sort of the great representation that we have on this working group. I want to highlight that we have 2 people from the library systems, from university library systems, which is a great perspective that we don't always get. We have a few people internationally, which is wonderful. We also have somebody who's representing a high school student population and thinking about research opportunities for advanced high schoolers. So just a really great diversity of perspectives. And next slide. So we have 2 objectives as part of this grant, and I'm going to present them in the order that they're in the grant, but we're actually doing them in the opposite order. So I'll start with our long-term objective first. We want to create a guide working group, which will actually be the educational working group, but we'll transition to kind of focusing on a accessible and universally applicable guide for FAIR data management, really speaking to all agricultural scientists. We're going to do that guide, which we'll produce in the 2nd and 3rd year of this grant. We'll do an overview of the current database environment, really focusing on AgBioData databases. The set of FAIR data management published standards. So this is where we're going to need to hear from the other working groups. If you guys have standards, of course, you'll probably be publishing them in your own white papers, but we can help build educational curriculum to make sure that those standards are well explained and getting out to everybody who might want to use them. And we want to write specific instructions for handling common types of data. So really trying to speak to the majority of use cases for our graduate students. So yeah, like I said, that'll be timeline a little bit later because we want the working groups to get some of those standards written. And we will be very excited to work with you guys on that. Next slide. This is the aim that we're actually digging into right now, which is going ahead and creating foundational education curriculum that can be incorporated into graduate student courses. Despite the fact that the grant says graduate student courses, I'll address that in a little while. I think we're actually gonna have a bit of a more expansive vision, but you know, develop training and history, importance, and current best practices for FAIR data science. And for this group, we're really interested in genomics, genetics, and breeding data.
I think that's a recurring theme we keep returning to. We can't have nice things, and this is why.
Oh, I think somebody was unmuted, but maybe they're muted now.
I want to remind everybody to mute themselves. Thank you.
Yeah, I thought it was a question. I was like, what? Okay, so the history and structure of GGB databases. We do have a long history of storing agricultural data into databases that have been around a really long time. So that should be part of our educational curriculum. And really how they have benefited the scientific community and how they continue to benefit the scientific community. So that's where we've started. Next slide. So we have this really diverse group who's interested in education. And basically we immediately discovered that we have a much broader vision of who our audience should be than just graduate students. So we've got somebody representing high schoolers, we've got people at majority undergraduate education universities, we've got people who really are interested in graduate students. I teach a graduate course that would cover this type of material. And we had somebody else who really highlighted the fact that postdocs and faculty need this as much as anybody else. So all of that, it becomes evidence of that we really want to think about all of those audiences. Also, there's a high value of practical experiences in this group. We want not just slides or concept papers, there may be laboratories, something you can do on your computer, biocuration. How do you actually get in there, practice submitting data, practice curating data, use the tools in the databases? So we really want that to be part of our educational curriculum. We do want to see publications coming out of the working group as well. So helping to disseminate this educational material that we're going to build to other people. And we're not completely dedicated to only agricultural databases. We think genetics, genomics, and breeding could really kind of span a larger gamut, thinking about TARE and some of the model organism databases. So we want to make sure we have a wide lens. Another important discovery we made early on is that there is a lot of fair data curriculum already out there. I would say the European continent in particular, they've got some really fantastic working groups across Europe that are producing beautiful fair data curriculum. And our job is not really to reinvent the wheel. I think if great curriculum is out there, we want to compile a list of those resources and share it with everybody in this network, but we don't want to redo it. So our solution has been to focus on the database aspect, which is much less well covered and particularly thin for agricultural resources. So that's where we'll focus our efforts. Next slide. So like I said, our timeline is a little reversed from the original objectives of the grant. We're digging in on this curriculum right now. We will be trying to get that wrapped up by the end of year one, including the practicals. We also want to produce an annotated list of databases for AgBioData that we can kind of use as a basis for how to build our curriculum and make sure we're including everybody who wants to be included. Year two, we will actually be able to teach our curriculum somewhere in person. We haven't figured out where that will be, but that is very exciting. And we're really enthusiastic to pilot it, maybe do some pre and post surveys to support our publication effort, and turn that into a white paper. And then that is the year that we'll transition from the education working group to kind of the guide working group, thinking about this fair data management guide. So we'll be digging into that. And starting in year three, we'll really want community feedback on all our products. Community, the other working groups can feed in their standards and outputs, and we'll turn that into educational material. And then finally publish that guide. Okay, next slide. We found a great paper that I would encourage everybody to check out, Fostering Fair Data Practices in Europe, by Claudia Engelhardt. And it is a lovely paper. It is also like 200 pages. So as a model, we're going for something that is similar in structure, but probably not quite as long. We'll see how ambitious we get. So next slide, I'll tell you a little bit more about the structure. We want to start with our motivation for doing this work and an introduction to biological databases. Again, like that foundational who, what, why. How do databases support fair data, such an essential part of making data fair, and maybe a brief history. From there, we really want to define the skills and the competences that we want to outcome to come out of our educational material. This is where we'll have separate competence profiles for different levels of learners, from high school all the way to doctoral level. And we want to define our learner outcomes. So really rigorous educational material we're shooting for here. Next slide. Here is our current structure of lesson plans we kind of want to develop. This is a bit ambitious. We may scale this back or we may expand it, depending on how much the community wants to dig in. I guess I won't read all of these to you, but we would be really happy to get feedback. We're thinking about FAIR, really practical finding, submitting, and obtaining data. Trust principles have been published for databases. We're going to talk about those. We'd like to dig in a little bit on the technology. How do databases work? How do websites work? What are APIs? And then since we do have 2 librarians, I think it would be great to talk about library resources, And then we'll definitely do a section that's very specific to agriculture. Next slide. The lab practicals are still a bit up in the air. We're curating a list of existing ones. So curation challenges, anything that might help students engage with databases. We may end up building our own. We haven't really decided, but that practical experience is a high priority for us. We will put resources for further engagement. And an example of that might be tutorials, because we know a lot of our AgBio databases already have tutorials on how to use them and how to contribute data. So we'd like to make sure that people are finding those resources. Okay, next slide. If you join the breakout session, here are some seed questions. We're really wanting the community to give us some feedback on agricultural databases. What type of learning do you think needs to be going on? Where would your students benefit most? Where would your postdocs, where would you benefit most from learning modules? What types of data are really your priority? How, is submission a priority? And just understanding what kind of classes or training are already out there that we might be able to leverage or build on. Final slide. So we do have a few questions in the follow-up surveys for the whole AgBioData network. Your input is so important to us and is really going to help guide where we're going with this. So we appreciate you taking time to fill out those surveys when they arrive in your inbox. We're still recruiting new members. We'd like to have some students. We currently don't have any students, and I think students telling us what they want to learn would be really great, right? Like to get them engaged and figure out what are they learning and what do they want to be learning. So if you have students who need a side project, sign them up. Or if you are a student, you need a side project, sign up. And feel free to join our Slack channel. We would love to hear from everybody. And thank you, that's it. I'm sorry for the early technical difficulties.
Anna-Rita, you're muted.
Oh, thank you, Monica. Thank you, Meg, for the presentation. I have one question for you regarding, like how the best way to kind of, there are already like many white paper or, tutorials, like let's say like book for fair data, but I was wondering if there is another way that we can also use that it's more familiar to students or to young generation. For example, I have always, now it's very popular that there are podcasts and people also listen to that to learn about papers or any other way, like also to learn stuff or new skill. Do you think that can also be a tool that can be explored for this topic?
Yeah, absolutely. We're super excited to think about producing videos, producing, I love the way that the software carpentry and data carpentry give you these easy to follow tutorials. They're still like written out, so you can kind of like Google your way into them. You're like searching for something and you land in the middle of this teaching resource, but it's very like step A, step B, so really practical. So I think all of those ideas are on the table. We would love to explore those.
Yeah, so there are two questions for you also. Michael is asking, what about workshop courses material for people already doing this work in private industry settings?
Yeah, we have not talked about engaging with industry at all, but that is a great idea. And if there are workshops, courses, or materials that you know about that are already out there, we would love to build on them. Again, like part of our first working group meetings, we're just like, let's figure out what's out there because there's so much out there right now, and how can we contribute instead of redoing? So yeah, if you have any information on that, I would love to hear more. And if there are industry partners who would be interested, we'd love to have a representative on the committee, or just to hear from them on what they have versus what do they need.
Yeah, and then, well, Monica just suggests you may want to look into the Genomic Education Partnership for lab material practicals. But yeah, so Thank you again, Meg, for your presentation. And I want to invite all the people that are joined today, that hearing about what is your experience in learning how to share fair data, or if you have attended any course, or if you didn't attend any course, which is the way that you will prefer to be educated on fair data. And with this, I want to invite Sarah, which is our next speaker. She will be presenting about the genome nomenclature work. So Sarah, go ahead and share your screen. Okay, here you go.
Okay. It's working? Great. All right. Hi, everyone. I'm here representing the Gene and Genome Nomenclature Working Group. So our motivation is that the importance of having accurate and persistent identifiers for genome assemblies and for gene models that are in the public domain. And if we have this kind of working, then this will help users understand where you've got multiple assemblies for the same species and multiple annotations maybe on those assemblies as well, which as we're entering the kind of pan-genome era where we're seeing a lot more assemblies of different individuals of a species, this is becoming more pressing. With the right kind of nomenclature, this will help users to replicate results and understand when they see differences in results, is that because of this kind of differences in assembly and annotations that have gone on before. We also want to help kind of orient users when they're comparing gene models across assemblies. And of course, for the groups that have gone to the effort of developing these resources, for them to be able to kind of track the citation and the downstream use of the resources they've made. So that's our motivation. And our objectives, well, we thought it was best to start with a glossary of relevant terms so we all know what we're talking about, which I'll touch on briefly. And then we're looking to develop guidelines on genome assembly and annotation nomenclature, in particular gene model identifiers, which I'll go into more today. And we're also thinking about genome QC metrics. So before we begin, to check that we're all thinking about the same thing. So when we talk about gene names, which we're not going to be talking about much today, this is when you have the kind of biological function assigned, like the reduced height 1 gene name. And then the gene symbol is this kind of abbreviated version, the shorthand, usually like 3 letters. So the RHT1 here. Then a gene model, which is what we're going to be talking about today, is where you have the kind of genomic feature that's been annotated on a genome and is predicted to produce a certain product. And that will have an identifier, which is something like here, which is kind of impossible to say, but very specific. So this is the kind of thing we'll be talking about later. And we're also going to touch on pan-genes, as I mentioned, kind of moving into the pan-genome era. And so a pan-gene is what we're considering to be a gene model, which is predicted in all the assemblies that you have for a given species or within your pan-genome. And again, which appears to be producing the same product. So just to kind of keep those in your mind before we move on. So in terms of genome or assembly naming conventions, there are some typical components which are often included. So you'll often have a species identifier, assembly version. You may also have the cultivar, accession or individual name kind of in there, and possibly also the group identifier of the group that actually did the work. So here's an example from the Darwin Tree of Life project of how they're naming their assemblies. So we have, this is the species identifier shown here in red. So this is where they have this Tree of Life identifier prefix, which for fish is F. And then for this fish species, they then take the first 3 letters of the genus and of the species, and kind of that's your species identifier. This is for the 3rd individual that they've done from this species, and it's the first assembly version. So these are the kind of different components that go together to make the assembly identifier. And we would like to identify best practice recommendations for AgBioData communities. When we're talking about the gene model IDs, the components that may or may not be included are, if you've got a polyploid, you might need a subgenome identifier in there, or want a subgenome identifier. You might have a chromosome identifier. You usually have a kind of an entity type. So if we're talking about a gene, it's often kind of G for gene, T for transcript, P for pan-gene. Is kind of so you know what type of thing you're describing. You'll have a numeric identifier numbering your entity. So for instance, if you have, these are often ordered with gaps. So what that means is if you start at kind of gene 1, chromosome 1, and then you go all the way down to your final chromosome and your final gene, and then you number them in order, And often they will have gaps. So each gene will kind of be an increment of 10 numbered. So if you can see down here where we've got the S identifier at 010030, the next one will be 010040. So you have this spacing, and that means that if another gene gets annotated in between two existing genes, you can give it a numeric identifier without disrupting the kind of the flow of all the IDs. And often there'll also be an annotation version as well to keep track of that. And so these will be put together typically in this kind of an ID, but I'll show you some more examples in a moment. So one of the things that's immediately kind of apparent and confusing is that you might want to capture the isoform of a transcript, the annotation version of the gene models, and also the assembly version, and you want to know which version of which is which without kind of being confusing. And if you put all of this together, well, for one, you get very long identifiers, but they are human readable and they are accurate. And ideally, we would want them to be machine readable too. And so here's some examples taken from the plants, different plant communities. And the first thing that you can see from all the examples is that they're pretty different. If you kind of broken down the identifiers into these different components, and you can see that not all of them are shared. So whilst everything does have an ID number, and something on species, that's kind of it. The rest are variable. Even whilst most of them have chromosomes, maize doesn't. And then within each column, how you represent the species is quite different as well, because you can either have with the one extreme, you've got Zea maize here with one letter for each. And then for this one is Brassica napus, one letter and two letters. Glycine max, you've got a 3 and a 2, Hordeum Bulgaria 3 and a 2, Triticum aestivum a 2 and a 3. So how you kind of balance these, or what is a convention within your species, and should this be standardized across all species to make it easier for people to understand which species we're actually talking about. And then there are special characters. So again, you can see here we've got some with dots, some with underscores. Sometimes you see dashes. Letters and digits are safest when it comes to machine readable. Dashes, full stops, and underscores might cause unexpected outcomes for the software that's consuming them. So what we want to do is look into guidelines on the best practice for identifiers. What are the kind of components to be included, conventions to follow, and the order in which you can put those together. That will be kind of easy for people to orient themselves when they come across these style of IDs. But the main kind of important question to address first is, do we even need IDs like this that are human readable? So some people don't think that's the case, some do. So it'll be interesting to hear your thoughts if you can join our breakout room and discuss. But just a couple of examples here. You can the Global Alliance for Genomics and Health have a system where they will take the whole of a genome sequence and compress that into an MD5 checksum, which of course is super accurate, because if you have even a slight change in the genome assembly, it will give you a different value, and you know that you're not talking about the same thing. But of course, you lose all that human metadata presented in that name. And the other option would be DOIs, of course, which I'm sure everyone is familiar with. So yeah, we'd love to hear your thoughts. The final topic, which I'll mention briefly because we're very much at the early stages of thinking about this, but we're also interested in quality metrics for assemblies and annotations. So although this is a step away from nomenclature, we feel quite strongly it's very important to understand and compare the quality and the completeness of the genome assemblies and annotations being produced. So at the moment, we're cataloging common existing QC metrics. And the thing to keep in mind is that older metrics, which were generated when assemblies were typically less contiguous, a lot of it was about kind of length and contiguity, which in an era where we're getting more telomere to telomere assemblies may be less relevant, and there may be other things that we need to kind of put more emphasis on. So we want to recommend a minimum set of metrics for comparing assemblies and annotations to each other. This is a list of our current group members. We're quite heavy on the plant side. So if we have people from the animal livestock kind of communities that would like to join us, you're very, very welcome. So Neil is our chair, who'll be reporting at the end of our discussion group, and Kapil is our co-chair. We're open to new members. We meet twice a month. So if you've got kind of 3 or 4 hours a month to spare and would love to contribute to it, we would like to have you. We're kind of time zone friendly. We're trying to span between the US and Europe and Japan. So we're welcome, open to everybody. And then finally, for our breakout session, the points that we thought to bring would be exploring these ID components. What are we missing? Is there anything that we've not considered, which would be a kind of good addition? Are long IDs acceptable, or should some bits be sacrificed in just to make them a little bit more manageable? Do we even need human readable IDs at all? And which are your favorite QC metrics for assemblies and annotations? So please come and share your thoughts.
Well, thanks, Sarah, for the presentation. So Monica is saying in the chat, I don't know if you have heard about it, the AG100PEST project is following the TOLID convention for genome assembly nomenclature. Do you guys consider that this project, or do you know that? Yes.
Yeah, so yeah, that's all the kind of the big global kind of projects we're considering for the ways that they are doing that, because obviously there's a lot of work on ahead. We don't want to just focus on AgBio. We want to learn from everything. There's things going on in the human sphere as well that we can learn from kind of broadly. So we are trying to scope broadly and then bring it into because obviously there are some things that are more of interest in ag. Polyploids are an issue which you wouldn't get that in the human space and such. So we want to kind of bring the best of what everybody is doing out there, basically. But yeah, if you have suggestions and contributions, please share.
So well, I want also to remind everybody that me and Pankaj, we shared the link for the survey that the Genome Nomenclature has for their work, so please fill in. And then there is one question from Suk. She said, this will be a question for every working group, but do you plan to think about how to enforce these standards?
I guess that's coming to me first before the other working groups.
Yes.
I'll take first. Anyone else wants to chip in? First, we need to make the standards. So that's what we're kind of on, first of all. And then listening to kind of Meg's talk about education and outreach. I mean, there's work to be done there, first of all, to kind of publicize your standard and have it accepted. And then, of course, comes the more, I suppose, lobbying to try and make sure that journals and places that will kind of publish the data will recommend, this type of data, this is a standard that's either necessary for your inclusion into whatever resource or publication, and I think it's a process. And we're very much at the beginning stage for this for us.
Sorry, Leonor is also saying the inclusion of unambiguous locus identifiers in scholarly publication is really important, but complicated for authors who prefer common names. Will you have recommendation for decoding that for publishing?
So I guess when you mean common names, are you thinking of like gene symbols?
Yes.
Yeah, definitely. It's a tricky one because it's different communities coming together. The biologists will be thinking in gene symbols, and the genome with the genome centric view will be coming with the identifiers, and so meeting those in the middle. But you would need to have a mapping, some way to kind of map between. It won't necessarily be one-to-one, but to actually capture that mapping to try and disambiguate.
So Patrice is saying the plant cell and more journals are requiring authors to have lists of gene IDs, since gene symbols can be for more than one gene. I have another question, which comes from a person that I am still a data user. So I am someone that find a genome assembly and then use the annotation. But my question is, like, sometimes for some crops there are many reference genomes, sometimes there are not group collaborating with each other, and so they publish two reference genome, and then they have their own genome nomenclature. And I was wondering but then, for example, when we submit to NCBI, they rerun the gene annotation, and so sometimes they rename gene. So I mean, like, will be there also guidelines in some way like that will help data user also to say, okay, if you like how we can like say, okay, there are different reference genome, there are different gene annotation, like some way that we encourage people to use some type of gene annotation that follows standards. So because it's also very difficult for someone that goes there to use data and say, okay, I found this reference genome, but will be there a way like also to encourage people to say, okay, maybe you should look at the NCBI first and then go for that, you know what I mean? That's a very serious issue that I found, because maybe you say like, oh, I found a new gene, and then in reality it's not. It's just that there are 2 reference genomes there and you don't know about it. What do you think?
I think there's different parts to that. So I think, first of all, having a kind of clarity that these are different assemblies, different versions, different annotations on the same assembly. So that's the kind of first bit that people need to understand when that's the case. In terms of a kind of RefSeq is always best because it's standardized. I think there'll be lots of cases where there's kind of community annotations that have maybe had a lot of manual curation put in as well, or maybe into a particular gene family. So it's quite difficult to say, like, this is the method. There'll be different strengths and weaknesses. I guess part of that is what we want to do when we're looking at least into the QC kind of metrics, to at least give a kind of a broad brush, help people kind of where to start. I guess that's.
So if I may add, do you have time? Yeah, so I think following up from Sarah's reply is that I think it is also overlapping with the pan-genome group aspect, because we are seeing different references now being used. So it's getting into the space where pan-gene and pan-genome aspects are being discussed. So I think we have some similar questions in our survey. If everybody can fill it up, there are 66 people on the call. And I would love to have your feedback so that we can survey what the community is thinking about in that way. Thank you.
Yeah, so that's for sure. I agree with you that there are many for the pan-genome that actually is now going to present. I want also to say that I think it's also a matter of educating people, like where to find resources. And I think I see a really like very point of contact with education working group. And so as Meg was saying, like we have to educate on databases, how to use data. And I think also for that, there is also a very strong collaboration that can come.
Absolutely, absolutely.
Well, I want to thank you guys. So there are other people are still commenting in the chat. So if you want to answer and to their suggestion and question, please go ahead. And with that, sorry, I want to invite Gerard from the Pan-genome working group to share the screen.
Okay, thank you.
And present.
Oops, screen. There we go.
Yes, it's downloading. Yes, great. Thank you, Gerard.
All right, thank you, Anarita. So I'm speaking for the Pan-genome working group, and I think what we're going to do is start off with a poll question, and the main reason is because when everyone started to organize for this, it seemed like everyone was approaching the pan-genome from different perspectives. I don't know, Anarita, do you want to post this now or?
Yes, I'm going to launch it now. Thank you.
Okay. Yeah, because when everyone started to gather, there were different needs that people were looking for in looking at the pan-genome. And see, oh, they're coming up.
Yeah, people are answering. So
okay, I'm seeing.
You can also go ahead and then I will share the results at the end. Yes.
Okay. Yeah. So when you think of the term pan-genome, are you looking at all the gene models? Are you looking at an overview of all the sequences that encompass the diversity of something that you're looking at? Or are you looking at something more granular, looking at a graph that looks at how the assemblies were done? Because every genome that you're looking at has a lot of qualities that are associated with it. And then also you may be looking at all different sequence variations, or it could be something else. Most of the people in our group, I think, were more in the I guess in the plant area, but I think most of the literature that has been out there, that initially started was mainly applied to microbial genomes. But that as sequencing has increased over the years, that's going to change. I'm not sure if the do I share the
do you want?
poll? Yeah, you can
still,
display it.
It's still open, so should I close it? Should I close the poll, or do you?
I'll just end it. Well, it seems like as they're coming in, everyone is looking at all gene models and then all sequences that encompass diversity. That seems to be the top view of what pan-genome is.
Okay, I will close it, and then do you want to share the results at the end of the presentation?
I guess I can just, yeah, we can do that at the end as well.
Okay, I will end it now.
Okay. And just to go over, let's see, here we go. Yeah, and so the pan-genome working group right now, it's a little over a dozen people. Ethelinda Cannon is the chair of the group, and she's been doing a very good job in getting us organized. And I think we've had about six or seven meetings since the latter part of December. And we've had a few presentations on, and as I had said earlier, everyone comes from different areas, but mainly it's been plant genome work. And as we've made discussions within the group, we've sort of broken out into different subgroups, and each group has participants that are trying to address these different areas. The subgroups are what are pan-genomes good for? This is an area where you would want to see the variety of applications that people would be using this information for. And then also in trying to build the pan-genome views, there's a lot of documented and reproducible analysis pipelines. And there's been some presentations on how that's being done using some automated procedures using applications like Nextflow, or even the documented procedures that are associated with the practical haplotype graph. And then unusually, what we've also seen is, over the last few years, there's been a number of presentations which address the visualization of pan-genomes in different ways. And we've been trying to look at that. And then the other part is when you want to look at data from different perspectives, how do you represent this data? What kind of databases are made? And also this sort of goes to connecting with the other working groups, the gene annotations, the gene variation and such. And that would be within the other working group where we're trying to connect the phenotype data to the pan-genomes and to, on top of that, do analyses. As I said earlier, our group mainly is mainly comprised of plant people, but we would welcome other groups that are working on animals or microbes to try to interject some new thoughts on how we could try to build pan-genomes. There we go. And then, as I had said earlier, the key question is, what are the pan-genomes good for? And a lot of this is the information that will help drive the directions that we would move to in the future. In our meetings, we've had a few presentations, one on the practical haplotype graph, and this was done by Clay Burkett, and And then we had an overview of the rice work that was being done by Rajmi John. And these are just two examples, but one of the things that we, and in the slides that you'll get later, you'll be able to see, if you select the slides, you'll be able to jump to the other information that is available. Just trying to. Okay, messed up here. So we have a lot of, so we're trying to document all the different efforts that are related to pan-genome efforts. And this would be a resource where we could use this information to get different perspectives of how people are working with pan-genomes. For instance, with the rice work, Rajmi provided some information about rice, which at this time, rice is really quite considerable in its efforts, in that over 27 different species have been sequenced, and you're getting a nice perspective of how these genomes compare to each other, and these would be applied to a number of different areas of research, just trying to analyze genes and gene families, looking at gene evolution, looking for novel alleles, especially with a lot of the search for crop improvement strategies. This would be one way to do that. And there's a number of research being done to look at present absence variability within different germplasm classes, and then also to just look how the crops have evolved and how they can be used for crop improvement. There's the other, and then also from Clay's work that he presented, there's been considerable work groups that have been designed associated with this work, and there's been workshops that have been held, and this work has been applied to maize and rice, and Clay has been working on wheat in this effort. And this is another way of trying to assess diversity within the genomes. As said earlier, depending on the method that would be used, a number of different pipelines would be developed to run these procedures. For instance, if you were to go to the practical haplotype graph, they have a very specific pipeline that they use, and it's even been modified where it's available as a Docker implementation, and makes it a little easier for the end user to use. And also there's other pipelines in development using for one Nextflow. And then yesterday there was a presentation in the genetic variation presentation that actually showed implementation of that to look at diversity in using the SNPs in germplasm. And another part of this is, when you have all this data, there's a variety of ways to visualize the information. And one of the things that we're trying to do is assess the different ways in which the data is being used and analyzed. There's as we start to see different applications appear, we've created this draft where we're starting to look at the different presentations. The images aren't coming out as well, but a lot of this has been done for legumes. Uh-oh. Well, I guess the bandwidth of my connection is not as great, but this is a better page to look at anyway. For instance, panache, which has been used for initially used for banana, has also been incorporated from Dave Edwards' group in working with wheat, and this is a nice visual of how the presence and absence of genes appear within the different genomes. Likewise, using a graph illustration, you can actually see how the assemblies of these reference genomes that are put together change over time, or change with different assemblies. And also, I think this would be something that would be very interesting when you look at the metrics associated with the assemblies. Being able to visualize the different outcomes would also be a good view. Let me move this over. And then there's been some really nice work where for the Legume Information Service, where they're actually looking at the gene level, and they're looking at the changes in different assemblies. And the legume people in the working group in the breakout sessions could probably speak more to how this is being assessed. And then likewise, a very similar illustration is associated with the gramming efforts in rice and then also you can see that there's some similarity or phylogenetic relationships associated with the pan-genome maps. So as you can see, there's really quite a range of different ways in which this data can be presented. One of the efforts for the group will be eventually is to create a dashboard where all the different visualizations could be sort of handpicked from each of the research groups, and a decision on how they need to use their data, at what sort of level of granularity that they need to view it can be selected.
Gerard, maybe you have like 1 minute left. If you.
Okay. Sorry about that. There's also different ways of working with the data, depending on the type of application that is going to be used. Different types of data would be searched and archived. And this is something that would also need to be worked with the other working groups for the annotation and gene variation, where there are some standard data sets being designed. Let's see, and then also ultimately on how this data will be used. This is this is a platform to look at how the information will be displayed using the genotype, the phenotype analyses. And when you look at the subgroups, all these questions that we have put forth will be addressed. How would you use the pan-genomes? Are you, who in the group is planning to use a pan-genome, and how would you represent and visualize and access a pan-genome, and how in what form would you want to share that? And then also, how are the efforts that are being present within all these research endeavors, how will they overlap with the other groups? And I think, so, that will be the subgroup questions. I hope I didn't go too much over.
No, don't worry. Thanks, Gerard, for the presentation. I think we will leave the question for the pan-genome to the breakout room session. And also I will share with you the results of the poll, so you guys can also discuss that during the breakout room session. So with this, I want to share my screen for the last. So before going for the break, I want to just remind everybody that will open soon the breakout room. Today we have 3 breakout rooms. We have the education, genome nomenclature, and pan-genome. If you are not very familiar with the breakout room, you can join them by clicking the sign below. And then you will have a window opening and you can join any of them by clicking the blue button with the join. So I want to remind the host and scribe that the breakout room will last 45 minutes. I will send an alert that I'm closing the room 1 minute before. Also, you are co-host of the meeting today, so remember to download the chat of the room if you need that. And if you want, please ask people before. You can also record the breakout room. And with this, I want to tell you that we don't have so much time for the break, but the breakout room session will start in 3-5 minutes. I hope you guys have very constructive discussion in the breakout room. So I think now it's time for the short report for the working group of today. The first one reporting is the education working group. So Mag, if you want to share your screen or like just report, please go ahead.
Yeah, we had a very wide-ranging discussion. We had a couple of students in the meeting, which was great, and they gave feedback. You know, we don't want necessarily perfect examples. We want examples of what doesn't work, because that's often, you know, even a better learning experience than what does. And they want curriculum that doesn't just cover NCBI and Ensemble, but like gets into the community databases. And we talked about data management plans for graduate students as a way to get them to think about their own data. We had some great discussions. Oh, thank you, Jackie. You're amazing. I was like, how can I remember all this? We had some really great discussions about involving industry. We're training the people that industry hires. There can be a much more positive relationship there, even if they're not releasing their proprietary data. They have the same struggles with data, and potentially there could be some much better coordination there. Thinking about getting fair education and databases as a core competency in our graduate programs and our undergraduate programs. So not just making the educational material available, but making sure it's a core competency that people are actually integrating into the curriculum. We talked about ontologies are a little scary. Might need to focus on that one. We're really excited to work with that working group and make ontologies fun and exciting instead of intimidating. I think that's most of what we talked about. I have a personal lack of knowledge of breeding data. So we talked a little bit about all of the groups that could help dig in on breeding data and make that a better educational module than something I could probably write. And anybody else who is in the session, if you want to jump in with anything I left out, please feel free.
Well, thank you, Meg. And now it's the time for the Genome Nomenclature. I bet is Sunil that is going to report. So Sunil, if you want to share your screen.
Yeah, okay. Can you see my screen now?
Yes.
Yeah, okay. Hi, everyone. So I'm Sunil Kachanmane Raju. I'm the chair of the Genome Nomenclature Working Group. And we also had a very good discussion. We didn't get through all the questions that we planned to discuss. But as you can remember from Sarah's presentation, we are thinking about what components should be present in the gene model IDs, what components are necessary, and what are the things that we can get away without adding them in the ID. So I think that was the major part of the discussion we had. And some of the thoughts were having a lot of metadata might be useful and important for people working with the same species or within the same community. but for others who are working outside the community, those metadata within the gene IDs may not be as important. But when you think about translating work from one species to another, again, those things are important. So I feel like even within the conversation, we were not sure whether all the metadata that we talked about are necessary and should be included in the ID, or some of that can be removed. For example, one of the comments is, should we include information about genes, transcripts, or a pan gene? Because once you keep sequencing more genomes, maybe the concept of pan-genomes or the identifier might change. So those are some of the things that we discussed. And regarding some of the QC metrics for assemblies and annotations, so most of the like the metrics like the BOOSCO scores or the QC details, some of them are provided when people submit their paper for when people submit their papers for the publications. However, there are some things that could be also important, but that are not provided. So that makes it difficult for others who are trying to replicate this work or use this information. So we are thinking of coming up with a checklist of basic metrics that should be provided along with genome publications. So we're thinking about reaching out to the journals, but also it's important that the community, the reviewers and the editors also take this responsibility and ensure that the authors are providing these information when they submit their manuscript. So I think it's a collective responsibility of the journals which publish this work, but also us as the community members, as reviewers and editors, we take responsibility of maintaining this minimum standard. So I think our working group will come up with a checklist that will be helpful, and we'll send this out to major journals that are publishing this kind of work. And I think we also talked about coming up with a white paper and also about publicizing our recommendations. So as I mentioned, again, we want to come up with a checklist and then allow journals to use that checklist, but also come up with recommendation and then have a way to publicize our systematic nomenclature. I think we'll be working with the education work group to create tutorials and other materials that can be provided to stakeholders so that the genomes that are coming up after, so we cannot change much things in the genomes that are already present, but with so many sequencing going on for so many species, we think that this will be a very good recommendation for future genome assemblies and gene ID nomenclature. So Pankaj or Sarah, do you want to add in things that I missed from the discussion?
Oh, one thing in the end we discussed is that we don't want to be working in isolation because there are a lot of other genome consortia and projects that are community-wide or kingdom-wide that are already working on some of these standards. So we would love to work and get connected with them. Monica suggested that we reach out to the steering group too, so that we can be part of that conversation, bigger conversation, rather than working in isolation. Thanks. Nice summary, Sunil.
Yeah, and one last thing. Again, we are looking for volunteers to join our working group. And as Sarah mentioned, we have a workload of less than 3 to 4 hours a month, and we meet twice a month. So if you are interested, please let me know. I'll put my email ID in the chat. So if you're interested, you can join in and help us in our working group. Thank you.
Thank you, Sunil. And now is the time of the pan-genome. I think Andrew also is going to report. Please, Andrew, go ahead and share screen.
Okay, can you see this?
Yes, we can see it.
I didn't have time to transfer all of the notes into this format, but got a couple of our questions in here. We had a good discussion. Focused on first the poll results. Everyone agrees that there are many ways to think of what a pan-genome is, and you know that's a consensus that everyone thinks there are many ways to represent it. So we were guided by our experiences with our current set of tools. People are doing things that are like the low-hanging fruit, and being able to do things like assess presence absence variation among genes, among a closely related set of genomes, and also want to classify genes as core, dispensable, or unique, depending on whether they're present in all of the genomes that you're focused on, or a subset of those, or unique to a specific genome within that group. And the other thing that pan-genomes of that approach are useful for is you can identify differences in annotation pipelines. I'd spot artifacts either in the assembly or in the annotation itself. Another need is that GWAS analyses really need to transition from a single reference to a pan-genome style approach. It's when you have a genome where maybe only 60% of the total set of genes are present in the reference, you may be missing a lot of things that are only present in other varieties. Another thing to look into as people go forward is they want to look at comparing the regulatory regions in addition to the genes and perhaps the impact of transposable elements on the regulation of genes. Yes, people are planning to make pan-genomes. Mostly everyone wants to do some pan-gene analysis, and this is going to be happening in a variety of species. Also, people are looking at practical haplotype graphs and other graph-based implementations. These didn't quite make it into this slide deck, so I'm going to switch to my other tab. Here, do you see this one? This is our notes document. The third question we addressed was, how do we represent or visualize things? We covered this a little bit in the presentation earlier by Jerry. And most of the activity is, I don't know, we're looking at tools that become available and, seeing, like, are these practical to apply to my data? And that sort of relates to how are we representing our data, and can they be, is it a big lift to transform your data into a format that works with the new cool looking tool that's out there? So this was something that we think we'll need to work on, identifying standards for representation and for different types of pan-genome views. And that also relates to how do we share pan-genomes. So a pan-genome can be represented in different ways. So if you're just looking at variations, you could have a VCF file. That's a very solid standard. It's been around for a while. And then for graphs, there are other approaches, such as I think it's GFC. I don't know, different things that are out there that we'll be exploring more. Let's see. And there's definitely overlap with other groups because everyone is touching on these issues with multiple genomes. So we're open to cross-pollinating with other working groups. Oh, and there we are.
Well,
does anyone else want to add to that from our group?
Well, thank you, Andrew, and all the speakers for the presentation. So before leaving, with the final remarks, we want to take a group picture. We cannot be in person, but please, guys, if you want, you can wave. I will take screenshots. So thank you so much, really, for being here today. And yeah, so please, all the other calls, take screenshot as you as well. Just a backup. And with this, I want to share the last slides. And so this is a reminder for the working group that presented today that please try to meet in the following month and to like develop some strategy to implement the feedback that you received today. We want to hear about this on the next AgBioData monthly webinar, which will be in April 6th. And I hope all you guys will join to hear more about it. And so I want also to remind people that if you are interested in joining any working group, you can reach out to me at agbiodata@gmail.com, or you can also visit our webpage and contact the chair. All the contacts are there. And with this, I want to invite you for tomorrow, which will be the last day. We will have the ontology, the Data Federation Data Collection Working Group. And I want to also to remind you that when we will close the Zoom link today, you will be asked to fill in a survey about the workshop today. So if you haven't done it, please do it. We will want to hear about your opinion and your thoughts about the workshop. And with this, thank you so much for joining us today. It was very nice to have all of you, and I hope to see you all tomorrow. Thank you.
Thank you. Bye-bye.
Thank you.